Now, by now, all of you understand the basis of genetics. We understand that the genetic code is stored in DNA. DNA forms chromosomes. Chromosomes in eukaryotic cells are packaged in pairs. And we really have an appreciation that all of us, any organism, is a composite of both parents that originally came together through gamete formation to form a new organism. Now today in this section, we will look at genetic abnormalities. As we will learn later that genetic abnormalities, for the most part, result during the process of meiosis. It is during the process of meiosis that the chromosomes spread out and move in different directions, forming gametes. If something goes wrong, we have an abnormality in the offspring. Just to review, Mendelian genetics makes a major assumption. The assumption is that two genes, a dominant and recessive gene, on a pair of chromosomes control a given trait. As we learned in the previous section, this is not always the case. Now, the genetics that we will be looking at today do not follow necessarily Mendelian's simple two-gene concept. The genetic abnormalities we will look at today will be sex-linked traits, autosomal disorders, chromosome abnormalities per se, traits due to multiple gene functions. Here we are looking at a human karyotype. As you know by now, the human karyotype is made up of 46 chromosomes. 23 pairs. Looking at this karyotype, we see that the chromosomes are paired up, and we refer to this pair as homologous chromosomes. As you notice, the X and Y are not truly pairs. The X and Y determine sex. Two X's, we get a female, an X and a Y, we get a male. This shows you an electromicrograph of the X and the Y chromosome. Notice how much bigger the X is than the Y. Therefore, you might expect any sex-linked abnormalities to be more associated with the X chromosome than the Y. Because the X chromosome has so much more information that can go bad. This table shows us the types of genetic disorders that we will look at today. We have X-linked recessive disorders. We're going to be looking at autosomal recessive disorders. We're going to be looking at autosomal dominant disorders. We're going to be looking at a variety of chromosome aberrations. And we're going to be looking at a variety of aberration in chromosome structure per se. Now please remember, autosomes are all the chromosomes minus the sex chromosomes. So when we talk about autosomal disorders, we are referring to any chromosome except the X and the Y. And when we talk about sex-linked disorders, we are referring to either the X or the Y chromosome. Let's look more carefully at each of these types of genetic disorders. X-linked recessive disorders. Now, in X-linked recessive disorders, the defective gene is going to be on the X chromosome. The female may be a carrier. Predominantly shows up in males, and examples of these disorders will be hemophilia, color blindness, muscular dystrophy, and so on. If you look carefully, you should see the number 8. If you don't see the number 8, it's possible that you have a genetic defect that results in your inability to see the number 8. Now, in a sex-linked recessive disorder, such as color blindness and or hemophilia, let's look at a cross between a mother who is a carrier. Now notice the carrier is written as black X and red X. The red X indicates the chromosome that has a defective gene. Now, in this case, the mother is a carrier. She is not colorblind. Now, if you look at the father has a X, black X, and a Y. The father does not have a condition of colorblindness. So doing a cross between the mother and the father in this case, we look to see what is happening. The father will produce 2 gametes, the X and the Y. Both are absolutely normal. The mother will produce an X egg and a red X. The red X has a non-functional gene. And in the Punnett square that we're doing here, we can clearly see the probability of the outcome. 50% of the females are going to be carriers, and 50% of the males will be diseased. The male cannot be a carrier because once the red X appears, automatically there's nothing to balance it out. So in this case, we can see how a recessive disorder carries itself out. Now, this is a relatively mild disease. Suppose we were dealing with hemophilia, which is a very debilitating disease. How could you possibly get a female that's hemophiliac in this process? The only way you can get a female that's hemophiliac or colorblind is that the female has 2 X's, 2 red X's, I should say. The only way that can happen is if the father has a red X. Now, with hemophilia, it's very unlikely because the father would be hemophiliac, and a hemophiliac would normally not live to the reproductive state. Therefore, you rarely, if ever, have females that are hemophiliacs. Again, we are dealing with recessive genes, but in this case, the defective gene is going to be present on autosomes. The defective gene is going to be recessive. Both recessive genes must be present for the disease to show up. Carrier states can exist, and examples of this, as you can see, will be albinism, sickle cell anemia, and cystic fibrosis. To illustrate this particular genetic abnormality, let's look at sickle cell disease, an autosomal recessive disease. In this case, you can see that the cells do not look normal, and they do not look normal because the hemoglobin, a major important protein in the cell, is defective. And as a result, a disease state will result. Let's look at the autosomal recessive disorder using sickle cell as a model. To your left, you can see the genotype and the phenotype. 2 big S's, normal. Big S, little s, carrier. 2 little s's means 2 little recessive genes coming together, and you get a sickle cell disease. So if we do a cross between the mother to your right who is a carrier, and a father who is also a carrier. Keep in mind, they do not show any disease symptoms. The mother's a carrier and the father's a carrier. You can see the father will produce either a big S and a little s, and the mother can also produce a big S and a little s gametes. So these are the possible gametes that are produced by the parents. If you do your Punnett square, look at your genotype and look at your phenotype. Your phenotype is going to be 3 normals and 1 diseased, 2 little s's to your right. Your genotype, however, will be 2 big S's, 2 carriers, S and a little s, and 1 little s, which of course will be diseased. So this gives you a clear understanding how autosomal recessive disorders are carried out and passed on. Obviously, if the father has 2 little s's, significant differences will come up in the phenotype. And obviously, if the mother has 2 little s's, all of the children will be diseased with sickle cell. Will be diseased with the sickle cell syndrome. Now we go, again, we're looking at autosomes. Autosomal dominant disorders. Here we're looking at a defective gene that is dominant. The defective gene will always be present in autosomes, as we said, and a single dominant gene results in disease. There are no carriers, because once a dominant gene is present, it will override everything else and it will be expressed. So again, remember, there are no carriers in autosomal dominant disorders. Examples of this would be Huntington's disease, a severe disease of the nervous system, and polydactyl, which is a person that has, 6 fingers. Now let's look at autosomal dominant disorders. Look to your left and look at the genotype and the phenotype. 2 big H's, 2 dominant genes, absolutely, disease state. A dominant gene and a recessive gene, remember, this is a dominant disorder, diseased. 2 little h's, normal. So, if we do just to illustrate a couple points here, if we do a cross between a mother who is perfectly normal and a father who is diseased, but is diseased with only 1 big H and 1 little h. The father still has all of the symptoms. Looking at a cross, you can see that the father will make 2 gametes, a big H and a little h. The mother can make 2 little h's, and the end result will give you a phenotype of 50% diseased and 50% perfectly normal. The way epidemiologists often look at disease origin and trace pedigree is through a pedigree map. Now, if you look at this illustration, to your right, females are represented by circles and males are represented by squares. A carrier is represented by half shaded in either squares or circles, and a fully diseased is shown by full colored squares or circles. In this case, we're looking at an autosomal recessive abnormalities. An autosomal recessive abnormalities means that both recessive genes have to be present for the disease or the abnormality to show up. Now, if you start looking at the generations, we looked at the bottom, we see the little male with 2 little a's. He is obviously has the albino syndrome. Now, we start asking questions, we realize that his parents would have to be carriers. And we start looking further, we see that his mother had a sister who was an albino. Well, now we can start looking at this interesting pattern, that for his sister to be albino, the grandparents would have had to have been carriers, big A, little a. So as you look at this pedigree map, you can see how epidemiologists go about tracing disease states, especially if they know what disease they're looking at, and more important, if they know if it's an autosomal dominant or an autosomal recessive abnormality. Now we get down to chromosomal abnormalities per se. We know that a full set of chromosomes, often referred to as euploidy, has 46 chromosomes, in humans at least, 23 pair. Now, what happens when something is going wrong and we end up with an organism that has more than one set? This is not good. More than one set most often results in a miscarriage. Nature has a way of correcting mistakes. When the chromosomes somehow bounce the wrong way, when this happens, nature has a way of correcting this problem and basically saying that this situation is incompatible with life. As a result, a spontaneous abortion results. Most spontaneous abortions, let me say this way, many spontaneous abortions are due to polyploidy. Now we'll look at aneuploidy. In this case, we're looking at a situation where an extra chromosome is in place, or a missing chromosome results through the process of gamete formation. Examples of this would be Down syndrome, often referred to as TRIS21, Turner syndrome, X only without a Y or additional X's, Klinefelter syndrome, 2 X's and a Y. Now, when we look at abnormalities that are due to sex chromosomes, most often the result will be a failure of the sex organs to fully develop. So in chromosome abnormalities that we've just discussed, this results predominantly from the failure of chromosomes to separate during meiosis, in the formation of gametes, of course. Now we're looking at a Down syndrome child, and today Down syndrome children can live to be healthy to age 40. Many years ago, this was not the case. Let's look at a karyotype of a Down syndrome child. If you look at the chromosomes, we see that the chromosomes are absolutely normal in terms of homologous pairs, but when we get to chromosome 21, we see an extra chromosome. And it is this extra chromosome that results in the physical characteristics in the Down syndrome child. This shows us the incident of Down syndrome based on the age of the mother. Now notice that when a mother is in her 20s, there's only one chance in several thousand of having a Down syndrome child. But as the mother gets older, the chances of having a Down syndrome child increases dramatically. Now, why do you think this is the case? Why do you think that as the woman gets as the female gets older, the chances of having a Down syndrome child increases? Well, if you think of it, if you think through this particular question, first of all, we understand that the female is born with all of the eggs necessary for reproduction. As the female ages, so will the eggs. So as the eggs age in the ovaries, there's a greater chance of something going wrong at the chromosome level. And this is probably the reason why the mother that's older has a greater chance of having a child that's Down syndrome. As we have said before, the problem in genetic disorders, in chromosomal abnormalities, results at the meiosis level, when the cells go through the process of separating chromosomes and forming gametes. Looking at this model to your left, you see what happens when you have a normal pair of chromosomes. The normal pair of chromosomes go through meiosis 1 and meiosis 2, and you go from 2N to 1N. A normal set of gametes are formed. Now look to your right, and we can see how failure of the chromosomes to separate either in meiosis 1 or meiosis 2 will result in gametes that are abnormal. There are other problems that may arise, aberrations in chromosomes themselves. Now we're looking at things like deletions. A big chunk of chromosome breaks off during the process of meiosis. Inversions, exchanges between non-pairs, translocation, duplications. Let's look and see what's going on with chromosomes that start to kind of fall apart. To your left, you see what we refer to as deletion. During the process of gamete formation and meiosis and so on, we can see that the ABCDEF chromosome has evolved into ABC, and the D part is missing. This is referred to as a deletion. An inversion takes place when a chromosome is being formed, but rather than hold true to its sequence of A, B, C, D, E, and F, it goes to A, B, C, E, D, and F. Genetic has to follow a pattern, and anytime you mess up this pattern, problems will arise. Now we're looking at translocation, where pieces of one chromosome from one homologous pair gets transferred to another chromosome. So you end up with a complete screw-up, I should say mess up. Let me back up. You end up with a complete dislocation of the chromosomes, as you can see. And finally, sometimes you have a particular segment of a chromosome that gets duplicated. All of these things lead to significant problems. Here we have an example of Cri-du-Chat karyotype. This is a severe disease that results in a variety of physical and mental disabilities. Look at chromosome number 5. We can see that the homologous pair does not look homologous anymore. A piece of it somehow has become dissociated and lost. And this, of course, results in a child that does not have a good prognosis. Now, how do we go about preventing genetic defects? Fortunately, we have significant ways of dealing with problems like this. Now, one of the best ways is in vitro fertilization. Now, in vitro fertilization is a process where we can harvest the eggs from the female, and then in a test tube or in a petri dish, we can fertilize it with sperm and basically end up with lots and lots of little embryos. We then can take an embryo, which is nothing more than a package of cells that are dividing, as you see in this illustration. We can pull off some cells without hurting the embryo and assess and analyze these cells to determine if the embryo is normal. And here we are looking at some of these cells, and we are looking for the presence of a particular chromosome. Chromosome 21. Remember, Chromosome 21 is responsible for Down syndrome, an extra chromosome. And unfortunately, as you can see, all of these cells have 3 copies of chromosome 21, which means that this embryo will result in a Down syndrome child.